Thank you for the introduction. I'm just going to dive into it. So the presentation is separated into two parts. In the first part, we're going to investigate whether the predictions from a protein language model correlate to within-species fitness effects. In the second part, we're going to go a step further and investigate whether these predictions from the language model can be used to detect impactful variants for field traits. So normally we use Ghysels to identify impactful variants. The issue with this approach is that due to linkage disequilibrium, we can't really detect the impactful variants at a high resolution. And the other thing is that the impactful variant has to be present in the population. So that means you're going to have a large pool of potentially beneficial variants that you just don't have the ability to detect with the normal Ghysels analysis. So an alternative approach to this is to use comparative genomic approaches where you have genomic information from multiple species. So here you have an alignment-based approach where you take the reference genome from multiple species, align it, and then you get something like this. From this information, you can see that certain positions in the genome is highly conserved, which indicates that deviating from the consensus here is associated with a cost to fitness. From this information, you can have predictions about certain nucleotides or amino acids, whether they're going to have a positive effect or negative effect on fitness. The issue with this approach is that you're going to have large parts of the genome where you can't align to other species, so you can't have predictions for these positions. And that's where these language models can be very useful, because they don't have that issue. They're trained on millions of, in this case, protein sequences, and from these sequences, they learn the underlying grammar of the protein language, and that means that for a given protein sequence you have, they can calculate the probability of observing certain amino acid residues, and if they occur very rarely across species, the model will predict those to be deleterious, whereas if you have something that occurs often, they predict it to be beneficial. So just to summarize, you have the alignment-based approach, where you have the issue that you're going to have large gap in the genome where you can't have predictions, but you have the benefit of being able to have very different predictions down to nucleotide levels. But when you go to the protein language models, where you have, for example, ESM, you have the benefit that you will not have these large gaps in the genome where you can't have predictions. And also, interestingly, with the protein language models is that predictions is going to be conditional on the genetic context, meaning that if you have different isoforms, then you can have a prediction for each variant in the isoforms. And we're going to try to validate the use of these protein language models in sorghum. So the data we use is a diversity panel composed of 400 accessions. And as an initial test, we check whether the evolutionary scores as calculated by SIFT, the alignment-based approach, and the evolutionary scores from ESM, so the protein language model, how well they correlate to the allele frequency in the population. And we see here that ESM have a stronger correlation with allele frequency compared to the SIFT, which indicates that ESM probably corresponds, the predictions by ESM correspond better to within species fitness effect. But we're going to test that now. So our hypothesis is that for certain genes, we're going to have consistent selection across many, many species, and that's also going to be the case within our specific species here, sorghum. And that means that alleles predicted to be beneficial by ESM, they will occur at higher allele frequencies within sorghum than expected under neutrality. So to make it a little simple, we're going to test if derived alleles, so mutations, occur at lower frequencies than expected under neutral assumptions, or whether the alleles occur at higher frequencies than expected. And we can do that for all sites in the genome. And then we have here the proportion of variants or sites that occur in these different selection coefficient categories. And those selection coefficient categories correspond to highly deleterious or moderately deleterious, and so on. And for all sites, we see that a large proportion of our new mutations, they are highly deleterious. And also, somewhat moderate proportion is slightly beneficial. But the thing is, we're not really interested in the distribution of fitness effects across all sites. We are interested in how the distribution of fitness effects is for certain categories of SNPs, as categorized by the evolutionary scores we have for the protein language model. So here we have the most deleterious group as predicted by ESM. And we see here we have a strong shift towards highly deleterious fitness effect for this category. But if you move to the categories predicted to be neutral by ESM, that's also what we see in our species, that these sites or these mutations, they are effectively neutral. And in the final category, those predicted to be beneficial by ESM, they also appear to be highly beneficial within the species. So to summarize, do the evolutionary scores by the language model correspond to within species effect? And our results do suggest strong support that that is the case. So in the next step, we're going to go and see whether these variants that we appear to be beneficial are also impactful for the field traits. So our hypothesis is that when you have a large load of these putatively beneficial variants, that will also lead to an increase in phenotype performance. So, and our second hypothesis is that those variants predicted to be non-neutral by ESM, they will also belong to a different fitness effect distribution, variant effect distribution. So we have our baseline model over here, where we have the phenotypes. We have a factor that accounts for being enriched for many derived allele. We have the random genetic effects, and then we have the PCs to account for population structure. When we go to the mean partitioning model, we include this component that accounts for being enriched for many derived allele in a specific SNP category, as I presented before. And then we have the corresponding model where we have it for random effects. So again, P here is the number of alleles in a specific category. And we can do that for all different categories we have. And what we see here is we have the halo circles that are the permutations, and then we have the filled circles that are the unpermuted associations, and anything that is non-transparent is significant at the Bonferroni threshold. And we see here for grain yield, there is a strong association between being enriched for many predicted beneficial alleles and the phenotype performance. So when you have many alleles predicted to be beneficial, you also have higher yields. And if you have many alleles predicted to be deleterious, you also have lower yield. So that is interesting from a breeding perspective. But the issue can occur that you have one trait that is important for breeders, where you have an improvement, but you might have another trait like protein content, where you have a decrease in performance when you increase the number of putatively beneficial alleles. So you have a kind of trade-off, and that means for breeders they have to make considerations about what they're going to value in the breeding program. Then we also have traits where the variants predicted to be neutral have a significant association with phenotype performance. And finally, we're also going to have traits that are shaped by local adaptation, where we're going to have where the protein language models do not have an ability to detect impactful variants. And when we have these models, we also do see that we have an improvement based from the baseline, whether we have it as a fixed effect component or a random effect component. So incorporating evolutionary scores into your model can help improve your ability to better assess your germplasm. So to summarize, yes, evolutionary scores, they can be used to detect impactful variants, but the relationship that these evolutionary scores have to specific traits, they vary. And what is important to consider here is that these protein language models, they detect natural selection over evolutionary time. So when you have traits where artificial selection and natural selection, they do not align, then using these protein language models to identify important variants doesn't work. But if you have something where artificial selection and natural selection, they align, that's where you're going to have the benefits of using these protein language models. So to end the presentation, I'm just going to acknowledge my advisors and my collaborators and funding agencies. And that was everything.